Давайте. Guys, sorry, good afternoon. Sorry for some technical problems. So, and start collecting, please, to listen to the lecture about viruses that cause respiratory infections. Guys, start coming, start connecting. Guys, start connecting with the lecture. Guys, or Vijayram, good afternoon, you are the first one. So we'll collect everyone here. So today we are going to discuss the RNA viruses that cause respiratory infections. Why we are talking about mainly RNA viruses right now? Because if you remember, out of all families of viruses, we have just 6 families where we have DNA viruses and 13 families where we have RNA viruses. So it means that RNA genome is most common in viruses, twice as common as the DNA viruses. So that's why by now we are studying RNA viruses. And today we will be discussing the very important groups for human pathology. These are Paramyxoviruses with many diseases in it, and automyxovirus with just one influenza virus. And we'll start with Paramyxoviruses. They are similar with automyxoviruses due to the word mixo, right? Because it denotes a large group of enveloped viruses that attach to glycoprotein cell surface and on the mucous membranes. So, and on the basis of fundamental differences, we may distinguish 2 groups, automyxoviruses and paramyxoviruses. We start with paramyxoviruses. Here you can see the taxonomy of the paramyxoviruses. This is a bit newer that had been given 5 years ago, for example. So, and you can see here that the family Paramyxoviridae may be subclassified into several subfamilies. And these subfamilies is one Paramyxovirinae and the second one Pneumovirinae. And we have 4 genera in the first subfamily and 1 genus in the second subfamily. So, and in the first subfamily, we may distinguish the genus Paramyxoviruses with 2 viruses there. With the parainfluenza viruses type 1 and 3, serotypes 1 and 3. Then we have the second group, rubella virus. And there we have mumps virus, well known, the causative agent of infectious parotitis, and parainfluenza of different serotypes, different from the previous genus. So then we have genus morbillivirus with only 1 virus, that is measles virus. We also may have some new viruses there, for example, henipavirus. We won't discuss it very much because there's a comparatively new one. And then we have a next family, Pneumovirinae, with 1 genus pneumovirus, and these are respiratory syncytial virus. So now take a look at the structure of the paramyxoviruses. This is the enveloped virus, so you can see on the structure the envelope with the glycoproteins, and inside it has single-stranded negative sense RNA. So the virus is pleomorphic in structure, so it can be, or in morphology, sorry, but quite often it will be spherical. And take a look here. You don't see the box-like structure that you used to see. So that's because the capsid of this virus is not icosahedral but helical. Can you see here? The nucleocapsid follows the helical structure of the nucleic acid inside. And this is the simplified scheme, and this is the electron microphotograph. You can see here how the nucleocapsid that was spirally shaped here comes out of the virion, right? So when the membrane, when the supercapsid is disturbed, so when you can see inside there is a nucleic acid, right? And outside it is covered with tightly bound with RNA capsid proteins. So that really protects the nucleic acid from being disturbed, right? And also you can see here that the nucleic acid is covered with nuclear protein, right? NP protein it is called. Then also several other proteins abound within nucleocapsid. This is a larger protein. It is called L protein. This is RNA dependent RNA polymerase. But for its activity, the phosphoproteins are needed. So these proteins make complex. This is NP protein, phosphoprotein, and large protein make a complex that acts as RNA-dependent RNA polymerase. Then the envelope is lined from inside with the M matrix protein. And the function of a matrix protein is to mediate the interaction between glycoproteins here and the nucleocapsid. So there's one extra layer, like one extra layer. And now let's take this blue one is the navy blue. This is a lipid bilayer that is derived from the previous host cell during the replication of the virus. And finally, we have glycoproteins on top. And I would like, this is a simplified scheme also, and I would like to draw your attention to the glycoproteins. The thing is that these glycoproteins can be same in different types of the viruses, and others, the second type of glycoproteins are different. So altogether in each genus, we have two types of glycoproteins, and one type is the same in all Paramyxoviruses. This is so-called, the blue one, fusion protein. You can see it here. See, this is a fusion protein. This is a trimeric molecule. This fusion protein, take a look here, is found on all species of Paramyxoviruses, everywhere. While the second protein, here it is shown as HN, and here you can see that this glycoprotein is responsible for attachment with a cell receptor. So, and in different groups of viruses, Paramyxoviruses, this protein is different. We at least may have three variants of this attachment protein. NH or HN, this is hemagglutinin with neuraminidase activity. Then we have H, just hemagglutinin, or G protein. And if again we take a look at this taxonomical table, you can see that pneumovirus, respiratory syncytial virus, it has F protein, as the others for fusion with the cell membrane when virus enters the cell. And the second protein is G glycoprotein for attachment. Then if you take a look here, then morbillivirus and measles virus here, by the way, it is not divided into serotypes, has H protein, hemagglutinin protein, and F fusion protein, while the others, parainfluenza viruses and mumps viruses, as an attachment protein, they use HN protein, hemagglutinin, with neuraminidase activity. So it's better for you to recognize which type or which genus has which type of the attachment protein. So, and we have additional SH. This is not really glycoprotein, but this is very small transmembrane protein that is not exposed to outside, that can inhibit the TNF-alpha signaling. Now let's take a look at the proteins and genome of this virus. So you can see here, and this is the genome of the virus, and in the genome we can see the color, so that coincide with the color of the proteins that are made up of this, right? This is an NP protein here, and then we may have H protein, then we have F protein, then we have M protein. This is a blue one right here. And then we also may have the L protein. This is the polymerase large protein. And also the protein that encoded in this fragment, that is phosphoprotein and non-structural proteins. So altogether we have 1, 2, 3, 4, 5, 6 structural protein in this virus. Okay? And what is important? The RNA-dependent RNA polymerase occurs physically in a virion. So this is the enzyme that tightly bound with the nucleocapsid. Because as soon as this virus enters the cell, the minus sense RNA should be transcribed into positive sense RNA. So let's take a look briefly at the way of replication. So the virus may use the different receptors. And here the receptors are shown that are typical for measles virus. And we'll discuss today generally the only one disease, that is measles virus. So, and the measles virus with this hemagglutinin receptor protein, glycoprotein, binds with two types of the receptors, CD46 possibly and CD150, that's the most common, right? So, and they enter the cell by fusion. When the envelope of the virus will fuse with the cell membrane. How is it possible? The fusion protein that is here, it mediates the fusion over the envelope with the cell membrane because they are very same. The lipid bilayer that covers the virus from outside is an envelope. So it is derived from the cell membrane of the previous host, right? So that's why it is possible for them to fuse. As a result, the nucleocapsid helically designed enter the host cell. And then the mRNAs are produced, and the mRNAs are sent to the ribosomes, for the proteins translation. So the C and V, these are regulatory non-structural proteins will be produced, but also other proteins are produced, and the way of production is different from that, for example, of the enterovirus, because many types of mRNAs will be produced, one for one gene each, right? So it means all pieces of the genome will be transcribed separately and all proteins will be translated separately, not like polyprotein in enteroviruses, right? So then, the whole process occurs in the cytoplasm. They don't need to enter the nucleus. So mainly the RNA viruses multiply in the cytoplasm. And the only influenza virus and very few, also some very few, give us an exception. So the whole process occurs in the cytoplasm, and the mRNAs will be translated into several proteins. So some proteins, like protein that's supposed to be surface glycoproteins, undergo post-translational modification. It means the glycosylation of these proteins occur. And once they are glycosylated, they are transported towards the cell membrane and start to be exposed on the cell membrane. By the way, from this moment, this infected cell may partially act as virus because the surface is modified by the viral glycoproteins. It means if we have a neighboring non-infected cell, the infected cell can attach through the hemagglutinin glycoprotein to the non-infected cell, and through the fusion protein, the cell infected with the virus may fuse with non-infected cell. It means that the viral capsids can be transmitted directly from one cell to another cell without going out of the cell. That is a very important feature of the paramyxoviruses. Okay, then when the glycoproteins are expressed there on the surface of the cell, the replication occurs also of the translation occurs also of the capsid proteins and the regulator protein that help in the replication. So then the progeny RNA genomes will be produced because the positive sense RNA will be transcribed from the whole genome and used as a template to produce progeny negative sense RNAs. And then they will be covered with the capsid proteins produced also from particular mRNAs on the ribosomes. So we have the progeny virions with the minus sense genomes. Then these nucleocapsids, we call them nucleocapsids because the RNA is tightly fixed with the nucleoprotein and also the polymerase complex proteins. So these nucleocapsids can be taken towards the cellular membrane, and the membrane in turn will be covered from inside with M protein. So the newly synthesized nucleocapsids will be taken to the modified inner layer of the plasma membrane, and then they are released by budding. This is a very typical way for enveloped virus. So the catch a portion of the modified cell membrane, and it starts to be an envelope of the newly progeny virion. That is the very simplified scheme of the replicative cycle. Okay. So now, few words about the measles as a disease, because that's a really very serious disease, and it may cause very severe complications. So, measles virus causes infection associated with acute respiratory illness, but then it can be complicated usually with immunosuppression. That is the result of regular, usual acute infection. But then it appears that measles virus is neurotropic. See here, right? So it means that may cause the infection of the brain tissues, and as a result, as a sequela of the measles, it may cause acute encephalitis directly as a result of the acute disease, and it also may cause the so-called long-term sequela as a result of previous acute measles virus infection. Many years later, then they may cause this infection, may result in so-called slow progressing chronic disease called subacute sclerosing panencephalitis, SSPE. And here we can see the main receptors that are found for measles virus. So CD46 and CD150 are epithelial cell receptors. But then we also measles virus receptor on the brain tissues. Where is it? That's why I think. So that is the receptor that can be used on the brain tissues. So, and also those receptors can be found in the dendritic cells and macrophages, and these are CD46 receptors and slums CD150 receptors. So through these receptors, the virus may infect also antigen-presenting cells. Now let's take a look at the pathogenesis of the disease. So, measles virus is transmitted as an airborne disease, and by the way, it is highly contagious. So, and the index of contagiousness is much higher than for coronavirus, by the way. So, when we are talking about coronavirus, approximately one person may infect 2-3 persons. But when we are talking about measles virus, one person may infect 18 to 90 the average person. So it means this virus is much more contagious than the coronavirus, for example. Then when it is transmitted as airborne disease, it enters through upper respiratory tract, mainly by using SLAM, and these are signaling lymphocyte activating molecules as a receptors. So then these viruses infect also lymphocytes and enter the bloodstream. And it propagates in the body and spread through lymphoid tissues around the body. By the way, in this moment, the person is not infectious yet. So then, you can see here, so the measles virus infected immune cells appear to transmit measles virus to epithelial cells, and there and to various organs, kidney, bladder, intestine, airways, and their viruses multiply abundantly. And then finally, when they multiply abundantly, so when it replicates in the epithelial cells, it will be released with the coughing and sneezing from the airways to the susceptible individuals. So, and you can see that the respiratory aerosol from the case contains a large amount of the measles virus particles. So from this moment, and usually it takes around 7 to 14 days, from this moment the person starts to be highly contagious. So then, because this virus may infect lymphocytes through the SLAM molecules, so the immunosuppression is a very important cause of infant death, because as soon as the immunosuppression is developed, babies and children start to be very susceptible to other infections. So, and major hallmarks of this generalized modulation of immune function are lymphopenia, the result of the depletion of the T cells, then prolonged cytokine imbalance, that also cause the immunosuppression and can give rise to secondary infections, and silencing of peripheral blood lymphocytes. They cannot expand in response to ex vivo stimulation. It means that the measles virus may infect lymphocytes on any stages of their differentiation, even those that are silent, resting in the peripheral blood. So that's why the decrease of the T lymphocytes cause very severe immunosuppression. This is a normal, regular sequela of the disease. But then it appears that this virus may reach brains during the dissemination in the body through three pathways, or three potential routes how measles virus may get into the brains. So the virus may assess the brains through the nerve bundles in the olfactory bulb, and through the olfactory neuron and olfactory bulb, so they can be transmitted to the brains. This is the first way. The second way, the virus can replicate in the endothelial cells of blood vessels, and thus it may infect brain capillaries, and the virus particles will be released directly into the brains. And also, the measles virus may infect monocytes in the peripheral blood, and the monocytes may transfer through the BBB and cause the infection of the brains. That's how they may reach the brains. So if it occurs directly within the disease, so then we have a sequela directly as acute encephalitis. But then most probably it occurs and when the virus enters the brains, so it may start multiplication, but quite often the virus mutates. And mutation occurs mainly in the M protein of the Paramyxovirus or measles virus. And then if the mutation of M protein occurs, then the virus cannot assemble. So again, I would like to show you the picture. See, the M protein is very important in assemble of the virus. So if the M protein is not produced, there will be no lining of the inner layer of the cell membrane. So, and the nucleocapsids could not bud from the cell. So the replication in brains occur very slow, and that's why we won't see any symptoms for a very long while until all cells step by step will be infected by cell-to-cell virus transmission. So then when the symptoms appear, nothing can be done, and usually the disease is lethal. I mean acute, subacute, sclerosing, panencephalitis. And now we take a look at the cause of the infection. So virus enters the body through the air droplets. Can you see, and this in this photograph, you can see the possibility to inhale these particles. Then virus multiplies, and that's we discuss how it travel in the body, finally start multiplying in the upper respiratory tract and the epithelial cells. Then usually, as a very first symptom, the complex spots will appear on the mucous membranes. They resemble grain crystals or grains of salt. So this is a very specific pathognomic symptom by these complex spots. So the disease can be recognized by the doctor. So then the symptoms occurs then as the cough, coryza, conjunctivitis. This is called 3Cs, and photophobia, and then rash. It is called catarrhal symptom, and then rash begins to appear, and on approximately 14 days. Then usually when the rash appears, the other symptoms gone, and then rashes begin to fade, and then we have the recovery of the disease. And here you can see that the in acute infection, the oligodendrocytes can be infected, and they may cause persistency of it. Then the damage of the neurons and oligodendrocytes, and then the damage to the brain tissues and demyelination of the brain tissues. Okay, here we can see the graph that demonstrates the pathogenesis of this disease. So, and there's step by step how these stages of virus replication, and this step by step how the symptoms appear. So once they enter the blood, infect tissues, and then the catarrhal symptoms and complex spots appear, and then we have rash development. And here you can see that IgM will be produced first, and they coincide with the rash development, and as a result of this we understand that rash appears not because of the direct activity of the virus, because this virus is not cytolytic, but because the antibody mediated immunopathological reaction that caused the damage to the epithelial cells. And then IgG later on will be developed, but then starting from the rash development, we may have very long-term immunosuppression. So, and this is the profile of interleukins. At the beginning, the cell-mediated immunity will be developed, but then it will be suppressed, and predominantly interleukins 4, 10, and 13 will be developed. That will tell about the humoral immunity, but not cell-mediated immunity. So, but humoral immunity can hardly participate or be critical in the elimination of the virus from the body. Usually cell-mediated immunity is protective against this disease. So now how diagnosis of this disease is performed. Frankly speaking, the symptoms are very clear. So if we find the, if we catch the catarrhal stage and complex spots and then rash development, and this is a very characteristic. So we may not need the clinical confirmation of the disease. So cough, coryza, conjunctivitis, upper respiratory tract symptoms, and typical exanthemas are performed. Then, but by the way, if we have very severe cases with acute encephalitis or post subacute sclerosing panencephalitis, then the disease can be confirmed serologically, and also it may be confirmed by the histological examination of the infected tissues, where we may see the syncytial formation. This is histochemistry, the histological evaluation of the skin lesions or respiratory secretion. We can see syncytial keratinocytic giant cells. So that is the result of the process of fusing infected cell and non-infected cell. That's very typical. And also, if you take a look here, we may find anti-measles IgM antibodies to confirm the diagnosis. Or if you find the IgG, we have to demonstrate again back. We have to demonstrate the rising of antibody titers. For this, we need to take paired serum. So I would like to explain again. Immunoglobulins, usually these are first class of antibodies that appear. They appear and disappear very quickly. So if you find IgM, that is the very clear sign of acute infection. But then if you catch just IgG, so when the level is not high enough to understand, is it a previous disease antibody titer or is a new disease antibody titer? So that's why usually we take serum twice, at the beginning of the disease and 1 week later. So to demonstrate rising of antibody IgG titer. If you demonstrate rising of antibody titer, there will be no doubt this is a specific antibodies. There will be no doubt that this is measles disease. That is for diagnosis. So now I would like to tell few words about the only way how can we stop spreading of the measles infection. And this is vaccination. So vaccine is called MMR, where we have 3 live attenuated viruses: measles virus, mumps virus, and rubella virus. And this is the one of the most contradictive vaccines. Why? Because so on the West Hemisphere, they don't like live vaccines. But the only live vaccine may cause cell-mediated immunity development. This is a problem. So the killed vaccines cannot cause cell-mediated immunity, just humoral immunity. But then humoral immunity is not enough to prevent measles infection and to fight with measles infection. And quite a lot of people don't like that this is to use this live vaccine, but it is highly effective in decreasing the number of measles cases. You can see here, this is the point where measles vaccines was implemented into and added to the calendar of vaccination. You can see the dramatic drop of cases and dramatic drop in deaths out of the measles disease. So, and then because of rejecting this vaccination not so far ago, the previous year, it was an outbreak in the United States, the highest incidence in the previous 100 years, right? So, that's because a lot of people start rejecting MMR vaccination for their children. And it should not be done. And this is a very serious mistake to reject this vaccination. Because it starts spreading again. Then I hope you understand that measles vaccination is the only way to control this disease. So this should not be stopped. And with this, I would like to finish for Paramyxoviruses. And you will learn some more about some more myxoviruses when you read the text. And this is just an example. So we shift to orthomyxoviruses. So they are also myxoviruses and come from the mucous membranes. And in there we have just influenza virus. But then you know that influenza virus is a virus that able to cause epidemics and pandemics. So epidemics, what is the difference between epidemics and pandemics? I hope you understand the scale of the spreading of the disease. If it is spread through one country or continent, this is epidemic. But when it starts spreading from continent to continent, so this is pandemic. And the very first well-described pandemic appeared in 1918, that had been called Spanish flu. So it was not Spanish virus. Why it is called Spanish flu? Because in 1918 it was a World War the First, where a lot of different countries fight with each other, against Germany mainly. So, but then the countries that were fighting, so they keep the incidence of the influenza virus infections and deaths out of these infections as a top secret. Nobody wanted to tell how many people died of it. But the only Spain claim and published the results of the surveillance and demonstrate how many people died of influenza virus. So that's why it had been called Spanish flu. So, but you should understand that during the 1918 pandemic, approximately 20 to 40% of the worldwide population become ill. This is much more than corona by now. An estimated 50 million people died, and nearly 675,000 people died just in the United States. It was really very devastating epidemic. So then the second pandemic was in 1957 to 1958, and it was Asian flu. And you can see here the formula of this virus was different. You can see here hemagglutinin 2, neuraminidase 2, type 2. And here you can see hemagglutinin 1, neuraminidase 1. So different, right? And first it was identified as usually in China, because that's truly this is a place where a lot of virus infection starts. And Guangzhou, it spreads from there to Singapore and then Hong Kong and then United States and the rest of the world. And estimates, and the WHO estimates the deaths ranging from 1 million to 4 million people. And that time, the researchers usually learned how to deal with influenza virus, and this pandemic had been predicted. So that's why when it starts, virus was quickly identified, and even vaccine was developed by 1957. So it means we react very rapidly towards this infection, or researchers that time. Okay, so then the next one happened in 1968, and we call it Hong Kong, and the formula is different, H3N3. It was not very large. And one more happened in 1977. It had been called juvenile because mainly the young people had been infected, so it had been called age-restricted pandemic. And again, it happens first in Northeast China and then spread to other countries. So, and what is interesting, in 1997 in Hong Kong, the first documented infection of humans by avian influenza virus with a absolutely different formula. H5N1 occurred, right? And since that time, the outbreaks happen mainly in Asia, regularly within almost everywhere, and they happen quite often among poultry and then also cause number of human infection. Then this avian virus usually cause infections with very high mortality rate, but usually not many people are infected. So these are not epidemics or pandemics yet. But then in April, in this century, in 2009, the outbreak of absolutely new strain that had been called first new H1N1, and then it was called swine flu, and now it is called Californian flu. So, and on June 11, the WHO raised its pandemic level to the highest level because it had been spread very quickly first to Brazil and from Brazil to the rest of the world. And this was the virus that originate from the mixing of the genomes of different type of the viruses, human viruses, bird flu virus, and pigs virus. So it was virus as a result of, that happens as a result of genes reassortment. So we'll discuss it later on. Let's take a look at influenza itself. Again, this is enveloped virus. It has minus sense RNA genome, and this genome is segmented. Please pay attention on it. There's a very important feature of the influenza virus, minus sense segmented RNA, segmented genome. And on the envelope, we have two types of glycoproteins, hemagglutinin and neuraminidase. So, and influenza virus is a major determinant of morbidity and mortality caused by respiratory illness each year. Still, coronavirus is beaten by influenza virus yet. So then, influenza virus really responsible for millions of deaths worldwide. And the hallmark of this, there's a most distinguishing feature of this virus, is ability to mutate on one side and high frequency genetic reassortment of the viral genomes. So, and these two types of the genetic variations, mutation and gene reassortment, results in so-called antigenic changes, so antigenic variations of the viral surface glycoproteins. So if it happens, this virus escapes from the immune system control. So that's how the new pandemic strains can be produced. So, and out of all three types, A, B, and C, it may happen in type A only. It means this accumulation of mutations or gene reassortment occurs in influenza type A but not B and C. Why it is so? Why B and C? Cause they are antigenically almost stable, B less and C absolutely stable, and cause sporadic mild disease and or very rare type B also may cause some epidemics. Why A is highly variable? That's because A may infect not only humans, but it may infect aquatic birds, poultry, pigs, horses, seals, whales, whatever animals. So by passing through the different hosts, and the virus, for example, may be passed from aquatic birds to poultry and back to aquatic birds and then to the seals and then back. So when the virus changes the host, it mutates first. That may lead to accumulation of mutations, and also it may the genetic reassortation may occur within these viruses if they infect the same host simultaneously, and then new viruses will appear. Structure of the Paramyxovirus. Virion is of middle size, usually around 100 nanometers in diameter, and may vary in size and shape. What is the structure? So, genome is segmented, single-stranded RNA of negative polarity, and in A and B virus, they have 8 segments, and in C virus, we have 7 segments. Each segment cause the translation into single protein, but it is possible that one gene segment may give rise to 2 proteins. The process that underlies this is called splicing or modification of the RNA. So you can see here a table of the viral segments and proteins that are encoded by these viral segments. Да, Ванечка Ивановна? Да. А что? Нет, я не болею. Нет. Да вы что? С понедельника я иду. Я знаю, да. Они меня подключили уже. Ну, у нас же пересдачи бесконечные, так что, Валентина Ивановна, я иду в понедельник. Договорились. Спасибо, хорошо, будем. Давайте. Sorry. So here you can see the viral segments, right? And what proteins are produced out of these segments. First, and here you can see the picture of the virus. So first, I would like to draw your attention again. 3 proteins that occur bound with the nucleic acid will be responsible to formation of the RNA transcriptase complex or RNA-dependent RNA polymerase that used for RNA transcription and replication. My dears, I hope you know the difference. Transcription is converting from minus sense to positive sense or from positive sense to negative sense. And replication is copying of the positive sense RNA many times or negative, in case of influenza, negative RNA many times. So 3 proteins act together as polymerase. Then this 4th segment code for hemagglutinin, and it mediates the attachment to the host cell receptor. Then we have segments that code for NP, nucleoprotein that covers the nucleic acid, and the symmetry of the nucleocapsid is helical. And then neuraminidase protein that on the surface, this is a surface glycoprotein. These are tetrameric structure, and it has enzymatic activity. Now then we have seventh segment that give rise to two proteins during the due to splicing of mRNA. M1, this is the matrix protein. It makes the matrix, this is the matrix protein. It makes the inner layer of the envelope. And it again mediates the interaction between glycoproteins on one side and nucleoprotein and genome on another side. And M2 protein, this somewhere here, this is a matrix. But M2 protein, this is a small protein here. Can we see? Okay, somewhere we will see it. Sorry. So, and this matrix M2 protein, and it mediates the uncoating of the virus in the endosome. That's we touch later on a little bit. So then, and the eighth segment give rise to the non-structural protein that are minor components, and they are important in the export of mRNAs and transplantation of mRNAs, and it happens as a result of the splicing of the 8 fragment. So, composition of the virus. This is a lipid bilayer with the glycoproteins. This is M2 protein somewhere here that make the ion channel when virus will be released from the endosome in the cell. And the blue are glycoproteins. So, glycoproteins form spikes about 10 nanometer in length, and they are really very important. First, they are important antigens that determine the host immunity, and the antibodies against hemagglutinin protect us against infection. And these glycoproteins usually undergo antigenic variations and participate in influenza virus immune escape. So if we treat the influenza virus with the detergents, the detergents or surface active substances, they remove this layer, lipid bilayer. So the virus will be partially destroyed and will be not able to bind with a new cell. That's why usually the envelope proteins are highly susceptible to the treatment of detergents, alkalines, acids, and others, not like naked viruses. They are more susceptible to the disinfectants and detergents. So this is a matrix, and you know it important in morphogenesis of the virus. There's a ribonucleoprotein that bound with the nucleic acid to form ribonucleocapsid. So now a few words about classification of the viruses. So we may have one genus, influenza virus, that contains of types A and B, and influenza virus C, that contain influenza of type C viruses, that usually infect humans and sometimes pigs. So, division into types is based on the antigenic differences and also ability to perform antigenic variations. How do we name the influenza viruses? Usually, if we take a look at this or at that. So first, if it is, first we put type, then if it is animal virus, we have to write here from what animal this virus had been isolated. Then geographic area origin is given for human viruses and for animal viruses. Then strain number, then year of isolation. This is 1930, this is 1968. And then the very important description, most important description. This is the antigenic formula of hemagglutinin, or the hemagglutinin and neuraminidase. And in a type A virus, as I said, the genetic reassortment may occur because they have segmented genome that makes this reassortment very common. When a cell is infected with two types of the viruses, we call it co-infection, with two different types of influenza viruses. Then when they perform replication and many copies of RNA fragments are performed, they may mix with the parenteral gene segments. Then the new virus may appear with the segments taken from different types of the viruses. And this genetic reassortment may result in very sudden changes of the viral surface glycoprotein. Then it will be absolutely new virus with a different viral glycoproteins. So, and these new viruses may cause very severe pandemics. So, and if we take a look at the glycoprotein, so the 15 subtypes of the hemagglutinin may occur generally. They are described, and out of these 15, just 4 may be isolated from humans. So, and the function of hemagglutinin, this is a binding protein. Then this is the major viral antigen, and variability of hemagglutinin may help them to escape from the immunity. So it has a complex structure. So, and then this is trimeric molecule that is contained HA1 and HA2 dimers, right? And each forms a large globule, and here you can see the fragment that were responsible for binding with the cell receptor. And here you have a so-called stalk, the globule and the stalk that anchors this dimeric molecule in the membrane. And here in a stalk we may have fusion peptide, this one that when it is open due to conformation, it may give rise to the fusion with the membrane. We have 15 types of hemagglutinin, and the 9 types of neuraminidase are described. But only 2 of them, N1 and M2, are typical for human subtypes. So, and you know that N5 is typical for avian subtype. From not so far ago, 1997, the avian flu viruses with N5 subtypes were able to infect humans. Okay, and the function of neuraminidase, it works as sialidase enzyme. It has the ability to split the sialic acids from the receptors and on the surface of the human cell, and by these it may mediate the more proper attachment or facilitate the attachment of the viral hemagglutinin to the particular receptors. But also the neuraminidase prevent aggregation of the progeny virions released from the host cell. And if neuraminidase is not working, so then the virus will stay aggregated on the surface of the cell and it will not be infectious. So that's why both neuraminidase and hemagglutinin act as targets for anti-influenza therapy. We discussed the replication cycle, then take a look at the sites that are targets for antiviral therapy. So, but then before, we have to mention anyhow these two types of changes that result from the mutations, and that is called antigenic drift, and from genetic reassortment, that is called antigenic shift. What is antigenic drift? So as a result of passing from the virus from one human to another one, the minor antigenic changes may occur as a result of accumulation of point mutations in the genes that code for hemagglutinin and neuraminidase. So, but then it takes a very long while. Usually it takes around 10 to 15 years from the starting point from one subtype, so then to the point where a new subtype is developed. Then you will tell me how to know whether we have a new subtype already or we don't have a new subtype already. Then it appears to be a new subtype when antibodies against the initial subtype won't react and won't neutralize the resulting subtype. Then, since that time, this is considered to be a new subtype of the virus. But then again, it is done step by step, and all these steps occur in the human body. So, in all these steps, the virus knew a bit different type spread through population. Again, some mutations may occur, and again they spread through new batch of cases. So then finally in humans the final subtype is developed. So this is a very long process. And then we have antigenic shift. See? So it occurs very quickly. So the new subtype may be developed within a half a year, and that is associated with major antigenic changes. So, and this occurs due to the genetic reassortment that we discussed already between different types of the virus. For example, human and avian, avian and swine, human and swine. When the segmented genome of the virus reassort readily in the doubly infected cell. So the antigenic shift usually results in appearance of new subtypes very quickly, and the only A can do this. Influenza B and C viruses are not able to develop new subtypes of this type. So virus replication. So attachment to the surfacellic acid through the receptor located on the hemagglutinin globula. So I showed you, right? Then virus penetrate by receptor-mediated endocytosis, and a whole virus appears in the endosome inside the cytosol. But then M2 protein mediates the uncoating of the virus. Between it is a membrane protein, and the very low acidity is required, and that is obtained by activity of M2 ion channel protein, because it permits the entry of the ions from the endosome into the virus particles. So we have uncoating, and the segments of the virus will be 8 segments will be released into the cytosol. And then we have a process of then this RNA segments will be taken to the nucleus. My dears, this is unusual for RNA viruses. As I said about Paramyxoviruses, usually RNA viruses multiply in the cytoplasm, with some exceptions. This is an exception. These RNA fragments will be taken to the nucleus, and in the nucleus they will be transcribed into positive sense RNase 8 segment, right? So then the transcription occurs by viral specific RNA dependent RNA polymerase. But then why it could not occur in the cytoplasm? The thing is that the mRNAs of the influenza virus will be not kept, and the cap should be taken from cellular mRNAs. So it will be cut out of the cellular mRNAs and ligated with the viral mRNAs. Only then the mRNAs will be transported to the cytoplasm and to the ribosomes to perform protein synthesis. And step by step, all viral segments will be transcribed and then translated outside the nucleus. But then when translation happens, the fate of the proteins will be different. Some proteins that are boxed here, right? Can you see? So 3 that form polymerase and NP protein will be taken back to the nucleus. And in the nucleus, the replication of the negative sense RNA happens on the positive sense RNA. And then many copies of minus sense RNA will be combined with these proteins to form nucleocapsids. So the nucleocapsids are produced in a nucleus. And then they assemble and then transported towards the plasma membrane. And those proteins that form those segments that code for M1, M2 protein, when these M1 and M2 proteins are produced, they will be taken to the cell membrane and line from inside the membrane that is modified. Okay, sorry. We have to start with hemagglutinin and neuraminidase. So when these proteins are produced, they should be modified in the EPR. So they will be glycosylated and taken by Golgi apparatus and inserted into the cellular membrane. Then M proteins will be taken here to line from inside the inner layer over the envelope. And then nucleocapsids will be taken here and start the process of budding. Can you see here? Here's glycoproteins, then M protein lines from inside, and then nucleocapsids are taken here. So then virus buds from the host cell. See, here you have a process of the release, right? So, but then when the virus is released, so it stays attached to the sialic acids on the surface of the cell. And here the drug may, if you use, may inhibit the activity of the neuraminidase, so the progeny virions will never be cut off the maternal cell, so the infectious virion will be never produced. This is the drug oseltamivir that used, but so, and the target is neuraminidase. And if we are talking about this stage, the uncoated stage, there's M2 protein here, Also may be used as a target for antiviral therapy, and the remantadine or amantadine are the drugs, so they are similar, that inhibit the M2 protein, so the uncoating won't occur properly. So if M2 is blocked, so the viral genomes will be not released into the cytosol. So these two are the major drugs that work here and here at that stages. Now a few words of the pathogenesis of the disease. I think you know it not so bad, right? So it is spread by droplets and also by contact with contaminated hands and surfaces, just like corona. But then corona is not destroyed in the GIT, but influenza virus, we think, is destroyed in GIT. So cells of respiratory epithelium become infected and spread to, and virus multiply there and spread to adjacent cells. And then within next time, the virus will spread through the bloodstream, but usually it is not manifested clinically, but the infection of the muscle cells may result in the muscle ache. So the incubation period usually is very short. So the virus itself is not cytolytic, and it disturbs the respiratory tract, but do not affect the basal layer of the epithelium. But then, if as a result of these infections, streptococci, staphylococci, and Haemophilus influenzae may cause secondary bacterial infection. So that these pathogens will get access to the not upper but also lower respiratory tract, and they may cause secondary bacterial pneumonia. That's why quite often after the influenza infection, if we have some complications, the antibiotic treatment is performed, but not to stop the virus replication. Virus is not sensitive to antibiotics. These are antibacterial drugs. Antibiotics are prescribed to stop those secondary bacterial infections. You know, the symptoms are typical: chill, dry cough, headache, high fever, muscle ache because of the propagation of the virus in the muscle tissues. And then the systemic symptoms usually last not more than 3 to 4 days, and the respiratory symptoms for another 4 days, and then it's gone if there is no secondary infection. So the most typical complication is the pneumonia. Laboratory diagnosis of influenza. Quite often we may use so-called rapid tests. This is identification of the virus, usually in the nasopharyngeal swabs. And now it can be done by direct fluorescent antibody assay, or now it is done by immunochromatography most commonly. Also, polymerase chain reaction may be useful, but it is not really in common use because it is too expensive for using in the large scale diagnosis. Usually the immunochromatography is used. So, but then in the beginning of the pandemics, usually the epidemics, the isolation of the virus occurs because we have to fully identify the virus, whether it is a seasonal influenza, regular, that doesn't cause very severe harm to the population, or whether it is a new strain that may spread very fast, so the morbidity and mortality can be very high. So the isolation of the virus is performed from the nasal washings or throat swabs, and usually the best way to culture this virus is culture in embryonated hen's eggs. So now the one very good cell line is developed, and it is called MDCK line. This is for dog kidney cell line. So, and M is the name of the researcher. But still, the using of embryonated eggs is most common. The presence of the virus can be detected by hemagglutination and hemadsorption, and identification of the virus is performed with hemagglutination inhibition assay with the specific known antibodies, or hemagglutination, on hemadsorption inhibition assay. Serological tests based on the detection of antibodies also can be performed. Prevention and therapy. My dears, take a look here. This is a scheme of virus replication. And some places are shown where we may influence somehow or prevent somehow the infection. Of course, the best way to prevent the infection is to block binding of the virus to the cell receptors, cell adhesion. But the only antibodies are able to do this, right, specifically. So if we have antibodies by binding with the hemagglutinin, they will prevent the binding of this virus to the receptor, so the abortive reaction occurs. Then amantadine and rimantadine, you can see here, inhibit M2 protein that causes acidification of the endosome environment and cleavage of the viral proteins and fusion of the virus and the release of the fragments of the segmented genome. This is very important. And then also when the virus will be released outside the cell, see the process of budding after the replication and maturation and assembly. So the neuraminidase, and the preparation is called oseltamivir, zanamivir, or some other trade names. So it prevents the release. So the virus stay aggregated on the sialic acids of the host cell. So if neuraminidase is blocked, the virus won't be released and won't be infectious. So prevention. I hope you are all vaccinated against this year influenza virus with a vaccine that had been given at something like 2 weeks ago, right? So, because you need to get the flu shot before the flu gets you. So, and we have many different types of the anti-influenza vaccines. But the problem with them is that every year new strains may cause infections, pandemics, or epidemics. So every year the vaccine should be updated. So, and the hemagglutinins should be inserted there from the strain that are actual or very important within this season. So every season we perform special vaccines. We may keep the previous year hemagglutinins, this year hemagglutinins of type A, and maybe add the hemagglutininin from the type B that usually cause the seasonal outbreaks. But then there's a special, I would say, kitchen to produce these very actual vaccines every new year. That is the major problem with influenza virus. Because, for example, this year we heard from the researchers that totally new influenza virus may cause epidemic this season. So that's why we need to be vaccinated, not to get together flu and corona. With this, I would like to finish. And thank you for those who came and even listened and even heard something. So, and the next week topic is still paramyxoviruses and automyxoviruses. And my dears, the week after the next week, if we start studying offline, will be a control class for bacteria. Please start to be prepared for it. Okay? See you in classes, hopefully. Goodbye.